All right, everyone, we will go ahead and get started because it just hit 2 o'clock. Good afternoon. My name is Caitlin Armendariz-Shergill. I'm the Manager of Outreach and Engagement for the University of Arizona Health Sciences. I'm happy to welcome you all to this session of U Arizona Health Sciences Tech Talk Tuesdays. Tech Talk Tuesdays is a series of bi-weekly conversations about engaging thought leaders, experts, researchers, and healthcare professionals in conversations around the advancement of technology within the health sciences. We're happy to have Dr. Roberta Diaz Brinton with us today. She's the director of the University of Arizona Center for Innovation in Brain Science, and she'll be presenting Regenerating the Degenerated Brain Following Your Discovery, Not Dogma. Just a quick note, today's presentation will be recorded and will be posted on the UAHS YouTube channel. I'll send out a link to everybody that registered later this week to that recording. And then after today's presentation, we'll open up at the end for questions. You're welcome to submit those via the chat and I'll ask them on your behalf, or you can jump in. And with that, I'll turn the time over to Dr. Brinton.
Thank you, Caitlin. And thanks again to the University of Arizona Health Sciences Tech Talk Tuesdays for the opportunity to share with you some of the journey that we've been on to translate our discovery into therapeutics. And part of that journey has been following the discovery and not the dogma. So to begin with, I'll be presenting 3 case studies: Regenerating the Degenerated Brain, Women and Alzheimer's, and Preventing Alzheimer's, in which each of these projects have really had to advance science, push the barriers, go to the frontier of science, and be aware of dogma, but not defined by the dogma. And then 3 of the lessons, I think importantly for successful academic entrepreneurs, is to listen, to learn, and to lead. So what is the listening about? Well, for me, the listening has been listening to molecules, cells, systems, humans. I was very impressed by the book on Barbara McClintock by Evelyn Fox, who entitled, A Feeling for the Organism. And that book really resonated with me, that the systems that we are studying are really in a conversation with us. They're talking to us. That the hypothesis that we generate is a strategy to interrogate the system. And once basically to ask the question, but like with all good conversations, one must listen to the answer. And the answer oftentimes is not what you initially thought, but it is nevertheless the answer. So the systems, the humans, cells and molecules can actually have a conversation with you if you listen to the data. And learn from those conversations, from all of the above, and to lead. And for successful entrepreneurship and translation, it's critical to have a vision, to be guided by your mission, which is, for me, very purpose and people focused. So to illustrate that listening is a story that is from my days as a postdoctoral fellow, in which I had the opportunity to be a blind basic science observer on a clinical trial in women with Alzheimer's disease. And one particular participant in that trial, who I'll call Dr. A, taught me a great deal. She was staying at the Rockefeller Hospital, which was on the campus of Rockefeller University. And I would spend long evenings with her walking along this esplanade that traversed 2 city blocks in New York, in Manhattan, that were part of the Rockefeller grounds. And she would tell me very rich stories about her life as an Adlerian psychologist and about the feuds between Jung and Freud, and then Jung and Adler. And it was a fascinating conversation. And after one of those evening walks, I took her back to her room, bid her goodnight, and closed the door. And 30 seconds later, I knocked on the door and entered and asked, "Dr. A, do you remember me?" And she was such a lovely woman. And she said, "I'm so sorry. Should I?" She could not remember me for 30 seconds, and I've remembered her for 30 years. And that was for me a transformational moment in my career as a neuroscientist. And I had to know why. And I learned from Dr. A. And from that encounter, I learned it wasn't that she didn't remember me, it's that she never encoded me. I never became part of her neural circuits of learning and ultimately remembering. And that set me on a journey to try to understand Alzheimer's disease. It led me to my purpose and my mission, the listening. So this is the story. This is the advent of the story that began in about 1992 with this serendipitous discovery. And that discovery was that a molecule that we'd been studying, allopregnanolone, allo for short, ALLO, could promote nerve cells to undergo mitosis. And what this really was, these were cells, they were residual neuroprogenitor cells derived from the embryonic 18 rat hippocampus. But none of that was known in 1992. And so the question became, what do I do with that? It's like a completely serendipitous discovery, but which I thought was critical to understanding how the brain, how cells could regenerate, and specifically how nerve cells could regenerate. So I followed that discovery with science, and a lot of science, some of which I'll present here in summary form. But I think it's important when we think about discovery versus dogma, there's a reason why dogma exists. There's evidence to support it. However, when you want to challenge that dogma, it's critical that one takes that discovery and applies the most rigorous science that is reproducible by other individuals and other laboratories in order to make the case that dogma either needs to be revised or discounted, right? Or abandoned. So in those days, the dogma was the human brain, and nobody ever thought the Alzheimer's brain could actually regenerate. The dogma was that in the adult brain, there wasn't regeneration, and particularly if it was diseased and aged. So this is a summary of some of the rigorous reproducible science that we've done over the past 30 years, almost 30 years, in which we understand the mechanism of allopregnanolone, why it promotes neurogenesis, the mechanisms underlying neurogenesis, and the mechanisms underlying its safety, because promoting mitosis and proliferation, regeneration in the brain is a dangerous proposition. And gratefully, allopregnanolone, a neurosteroid produced in abundance during the third trimester of pregnancy, has worked out all of those critical issues to promote expansion, a fourfold increase in expansion of the brain during the third trimester of pregnancy, and to do it under controlled circumstances. We've also I'm hesitant to use the mouse because that tends to advance the slide. We've also worked out the time course for this process. And that turned out to be very critical to understand that activation of a membrane receptor, the GABA chloride channel complex, leads to activation of early response genes and subsequent transcriptional factor genes that ultimately lead to neurogenesis and the generation of new nerve cells. We've also worked out the optimal treatment regimen, and that turns out to be once per week. Typically, we think about therapeutics as everyday kind of administration, whereas for allopregnanolone, one really must pay attention to the regenerative time frame for the brain. So what we learned is that more is not better, that you actually have to follow the biology of regeneration in the brain. And ultimately, our work led to understanding and discovering the discoveries that allopregnanolone could promote neurogenesis, nerve cell generation, as well as the generation of new oligodendrocytes that promote white matter regeneration. And then because it promoted cognitive function, we knew, and basically learning and memory, going back to Dr. A, the ability to encode new information and recall that information, we knew that synaptogenesis had occurred. In addition, we discovered that allo promotes the bioenergetic system of the brain through regulation, promoting mitochondrial function, and also acts as an anti-inflammatory that promotes synaptic function. In addition, we discovered that allo reduces the burden of Alzheimer's pathology in the brain, very likely through mechanisms that promote cholesterol efflux. Collectively, the data indicate that allopregnanolone can promote the regenerative systems of the brain, as well as modifying the disease process of the brain. We knew, in addition, that we had to have a resident set of cells that have the potential, that have the possibility of regenerating. And in our preclinical studies, we had identified that essentially it's really during this early phase of Alzheimer's disease where allopregnanolone is most likely to have therapeutic benefit. This gives you a rough timeline of our whole developmental process for developing allopregnanolone as the first regenerative therapeutic for Alzheimer's disease. And you can see that it's quite complex. But one of the other reasons that I wanted to show this to you is that you can see that for the first part of this journey, there was essentially no funding. And I'll tell you a story about why that is. So I was a young assistant professor when I made this discovery, and I put in an NIH grant, was reasonable, and it came back that it would not be funded. And no one had ever educated me about NIH grants per se. And I didn't realize that if it was not funded on the first round, you could actually resubmit. So I took no for an answer. I put this project in a drawer for about 10 years, and then received funding from the Norris and Whittier Foundations, philanthropic funding. And that philanthropic funding allowed me to pull that project back out of the drawer and continue the research. And part of what that also did was allow this time frame, a substantial time frame, from when I first made the discovery and started working on the project to generate that preliminary data, to handing this project off to a very gifted postdoctoral fellow, Dr. Junming Wang, in my laboratory, and saying, here, see whether you can reproduce this. Different time, different hands, let's see if we can reproduce these data. And that's what happened and ultimately led to funding from the National Institutes on Aging, but also the Alzheimer's Drug Discovery Foundation. So one of the take-home messages is diversifying your funding portfolio, and I'll talk more about that later. The preclinical work ultimately led to our ability to conduct a phase 1b/2a clinical trial, and I'll talk more about what that means. Essentially, a first-in-human trial in persons with Alzheimer's disease. And because each of us is exposed to high levels, about the highest levels in the human during the third trimester of pregnancy, the FDA allowed us to treat persons with Alzheimer's disease for 3 months, which is what we did. We treated 12 women and 12 men, age-matched men and women, who had a diagnosis of either mild cognitive impairment due to Alzheimer's disease or early stage Alzheimer's disease. We were able to conduct our primary outcomes, which were safety. Was allopregnanolone safe in this aged population, well past birth or pregnancy, and whether there was any evidence for inducing what was called amyloid-related imaging abnormalities by MRI. And that turned out to be, at first, it was a bit of a frustration that we had to do this MRI testing, but it turned out to be an incredible opportunity for us, which I'll talk to you about in a moment. And we were able to establish the maximally tolerated dose in these individuals, as well as the what turned out to be the potential maximally effective dose for allopregnanolone. We also had multiple exploratory outcomes, one of which were these MRI-based surrogate biomarkers, cognition, and an iPS-derived strategy for potentially developing a biomarker of responders. Now, the reason why we were, I'll tell you about that in a moment, right? So the outcomes of the trial were that there were no indicators of toxicity, no indicators of amyloid-related imaging abnormalities. We were able to establish predictive pharmacokinetics. There was no sex difference in the maximum tolerated dose. There was a sex difference. I'm sorry, back up. There was a sex difference in the maximally tolerated dose. There was no sex difference in the maximally effective dose. It turns out that men were more sensitive to allopregnanolone than women. But importantly, there was no sex difference in the maximally effective dose. Cognitive analyses were feasible. The ones that we were performing in this group were feasible. The MRI surrogate biomarker of regeneration was feasible, and that it was feasible to develop an iPS-derived, an inducible pluripotent stem cell strategy as a predictive biomarker of responders. So what does some of that data look like? I'm sorry, I'm going to have to delete this, which didn't appear in the, oops. There we go. There we go. So on our exploratory biomarker for the MRI biomarker, the question we had was, if there was regeneration occurring, could we actually detect a change in that area of the brain, the hippocampus, that area of the brain that you're using quite extensively right now to listen, encode, and connect neural circuits that you already have established, but that may help you to remember some of the key points from this conversation. Okay, so the question was, could we detect a change in the volume of the hippocampus after just 12 exposures to allopregnanolone once per month for 3 months? And we essentially were able to detect an interesting finding. Here, what you see is the placebos who do not carry the risk factor for Alzheimer's disease, and those that are treated with allo. And you see that on average, allo has no benefit and no harm on these individuals who do not carry the risk factor for Alzheimer's disease. In contrast, allopregnanolone was able to, relative to the placebo of those individuals who carry the risk factor, the ApoE4 risk factor, the ApoE4 gene risk factor for Alzheimer's disease, allopregnanolone was able to stabilize hippocampal volume over those 3 months. Some did lose hippocampal volume, not to the same extent as the placebo, and some gained. Right? And on average, the hippocampal volume was sustained. Now, this was important because this was a key finding that was predicted by our preclinical, our studies in humanized ApoE mice, that ApoE3 carriers were not responsive, whereas ApoE4 carriers are responders, potentially responders. And that informed our design of our phase 2 clinical trial going forward. Oh, there's that slide. That's the one I was looking for. So in terms of could we predict responders, what we found was that indeed these neural stem cells derived from blood transformed iPS cells. So we took blood from each individual prior to their randomization into the trial, and we had their blood, and then we reprogrammed cells, their blood cells, into inducible pluripotent stem cells, and then differentiated them to neural stem cells and asked, do you respond to allopregnanolone? And in this case, we were looking at mitochondrial function. And then we did a correlation between their mitochondrial respiration and their change in hippocampal volume, and found a very high correlation. And I can talk more about that later in the questions. So this gives you a timeline of what we have achieved thus far, and our discovery and translational research, and our phase 1 clinical trial for safety and maximally tolerated dose determination, which had data suggestive of a response that led to the development of a phase 2 clinical trial, which we will begin enrolling in April of this year. While the phase 1 clinical trial was being conducted, we conducted a substantial amount of chronic toxicology that was required by the FDA in order to advance to a phase 2 clinical trial. And we currently are developing multiple routes of administration for allopregnanolone. Currently, we're administering allopregnanolone intravenously once per week, and obviously for substantial, broad clinical utilization, other routes of administration would be greatly advanced clinical use. So again, I remind you of the funding that went along with all of this, that it's a staged process, and at each stage of the process, multiple sources of funding are required. And one of the take-home messages for me as a young investigator, well, actually, I didn't learn this until I was an associate professor, but I teach everyone, all of my mentees, this lesson, which is to develop a relationship with the program officer responsible for that area of research. And the reason being is that when they know you and come to know you better, you have a potential advocate for this importance of your research. Now, of course, one of the things that I also learned about NIA program officers is that they're incredibly smart people. They know the field probably better than anyone because they have to constantly stay on top of the field. They are looking at grants that are being submitted. They have to understand whether these grants are innovative, whether they're advancing the field, whether they're conducting a very important research to reproduce a novel finding. So they make a lot of decisions like that. And part of their role is to come to know the investigators, because we are their team, right? That as NIH-funded investigators, we're part of that program officer's team. And they are evaluated on the science and the importance of the science that they manage. Right? So it's a great relationship, and you actually learn a tremendous amount from these program officers. They'll give you some of the most challenging conversations, scientific discussions I've ever had are with program officers, because they know a lot. Okay, case number 2. The dogma was 2 out of every 3 persons with Alzheimer's disease are women, and that's true. And so far, it is held up to be true. What the dogma also is that the only reason why 2/3 of all people with Alzheimer's disease are women is because they live longer than men. And on average, they do by 4.5 years. What our discovery research has shown is that it's not because women live longer than men. It's because they start the disease earlier. And they can start the disease in midlife. And that midlife endocrine and aging transition of the menopause can be a tipping point for the onset of Alzheimer's prodromal phase. We know that Alzheimer's, by the time someone is diagnosed with Alzheimer's, they've had the disease for 10 to 20 years. It's interesting, if some of you have been following, that Tony Bennett has acknowledged publicly his diagnosis of Alzheimer's disease that occurred in 2016. But his family was very aware that his cognitive capabilities were quite challenged long before the clinical diagnosis. And that's very typical. Again, Alzheimer's disease can be a 20-year prodromal phase to the actual diagnosis of Alzheimer's disease. And what our discovery research has shown is that these diseases, Alzheimer's, and I'll talk more about other age-associated diseases, are progressive in nature. And the reason is because the brain is changing. One of the other really fascinating discoveries that we've made is that the brain is constantly trying to survive, right? And it transitions through multiple stages of attempting to survive. And each of those stages actually are quite different from each other. They have common features, inflammation being one, dysregulated bioenergetic glucose metabolism in the brain is another, the generation of beta amyloid is another. They have common features, but the severity changes, and the severity is also associated with kind of the onboarding of other systems of biology. So that progression is one, if you will, one system is affected, and then 2 systems, and then the interaction between those 2 systems leads to a 3rd system being affected. So this complex, dynamic progression of disease requires that we think about therapeutics in a very different way than currently. The current approach has been to target the pathology of the disease, specifically beta amyloid deposition in the brain, beta amyloid plaques. And that has resulted in 99% failure. Changing the amyloid load in the brain has not resulted in an effective treatment for Alzheimer's disease. We can talk about the Biogen, trials later in the talk. But thus far, there has not been a single therapeutic, and there have been many in clinical trial, that target beta amyloid. Kind of the dogma in the field, targeting beta amyloid, has led to clinical success. And part of that is the target. But going forward, we really have to think about a precision medicine approach to Alzheimer's disease. How did that person develop Alzheimer's disease? Where are they in the stage of the disease? And what systems are involved? Coming to the issue around women and Alzheimer's, coming around to the issue of women and Alzheimer's disease. We embarked on a program of research, again, that has been 30 years in the making, of trying to understand why the female brain is at greater risk for developing Alzheimer's disease. And we started kind of in the aged animal, and we kept moving earlier to understand what was happening that we were detecting later in age, but was actually starting much earlier. And we started studying the menopause, animals that were menopausal, animal menopause, reproductive senescence. But the data, again, another serendipitous discovery, the data said, no, menopause is the end. It's the end of this transition that can start the clock on Alzheimer's. It was really during this endocrine transition of aging transition of the perimenopause. And the perimenopause is a bit like puberty, right? Lots of things are changing. And it was understanding that whole process of the perimenopause, the neurological process of the perimenopause, that led us to two critical insights. The first was estrogen is, I call the queen of Darwin. She has left nothing to chance in the brain. Estrogen promotes the entire glucose metabolic system in the brain, from glucose uptake to metabolism to ATP, oxidative phosphorylation, and ATP generation. And the loss of that estrogen results in about a 20 to 25% drop in glucose metabolism in the brain. And that sets off alarms. And the alarm is, I'm starving, that the brain is starving. And from there, many subsequent adaptations occur to address the energy deficit in the brain. The other, and so listening to that brain, all along the way, and that conversation with the data, hypothesis testing, and then listening to the data, was paralleled by clinical interactions in which I would be speaking with physicians about estrogen action in the brain. And invariably, physicians would ask me, well, what about all my patients who are taking these soy products for their menopausal symptoms? And I couldn't tell them the answer. I didn't know what these isoflavone soy products were doing in the brain. And it took me a while to actually convince the team to investigate this. But eventually we did. And we also learned in the course of this journey is that women are voting for their brain health with their breast health. The reason why women elect not to take hormone therapy, which we had shown to be effective in promoting estrogen action in the brain. The reason why women elect not to take estrogen therapy is the fear of breast cancer. So we embarked on a innovative therapeutic development program to incorporate what women wanted, which was a safe and efficacious, alternative to estrogen and hormone therapy, with the plant-based molecules that they were willing to consume. Now, what I will tell you is that these are typically over-the-counter products, which are not clinically tested, which are not they do not have oversight by the FDA. So we wanted to address not only efficacy, but safety. And so embarked upon a program of research that follows all of the same kinds of criteria that we have followed for FDA approval for example, allopregnanolone. And the bottom line is we've conducted many years of preclinical research demonstrating that first we developed a formulation of using computational chemistry as well as bench science, a formulation that worked as effectively as estrogen, 17-beta-estradiol in the brain. And we conducted from there, in addition, we did translational study analyses to document safety. And we did this over a 12-month everyday exposure, which is quite unusual in the academic field. But again, we were interested in safety. So we were interested in whether this formulation promoted proliferation of breast and uterine tissue in our animal models, and it did not. We then went on to conduct a phase 1 safety trial, and again, establishing safety and the maximally effective dose of our phytoserm formulation. And that's the chronics. We did this again for 3 months and essentially demonstrated safety again. And we are now working to, we just submitted a proposal to conduct a phase 2 proof of concept clinical trial of phytoserms for targeting brain glucose metabolism and cognition during the perimenopausal women. And lastly, our work has resulted in the development of an academic enterprise and entrepreneurship to develop a company, New Therapeutics, that will be responsible for moving our academic, our university-based discovery, translational and clinical science into the commercial world to ultimately reach the critical people in this endeavor, our target populations. So more on successful entrepreneurship. So coming back to those 3 properties of listen, learn, and lead. We as scientists are humans, and I listen a lot to my colleagues in my research laboratory. I took 2 photographs, one, pre-COVID, one, during our COVID lab endeavors, just make sure everybody's included. But it's critical that one learn leadership. And I have learned some leadership. I've learned a lot of leadership from my team members who are extraordinary, wonderful, amazing individuals. And part of that is that scientists are ultimately purpose-driven people. They may have articulated that, they may not have articulated that, but science, as you well know, is an endeavor that is not for the weak. And the purpose that we each bring to our science is critical. And communicating that purpose with our teams and inspiring our teams to understand that purpose and to take that purpose on for their own life, for the time that they are with you and potentially in the time that and beyond. So that's a skill set that I don't think we learn in graduate school, but is critical for successful entrepreneurial science, translational science. And I encourage each of you to think about what is your vision, and from that vision, what is your mission? And I find that approach certainly is attractive to young investigators, often young investigators, and to senior investigators. It's not for everyone. It's been very successful for me, and I encourage you to consider it. And learn as you go. I learn every day from everyone that is in the Center for Innovation in Brain Science, my team members, and from the patients that we interact with in our clinical trials, and from their physicians. When my most successful science has been when I listened to those humans and their physicians and brought those questions back to the bench to try to understand what are the underlying mechanisms to their questions. So I encourage you to give that some consideration. And then again, it takes a team, it takes a village, a very large village, to do this kind of science. And with that, the financial and intellectual support from multiple sponsors that I've been fortunate to have, that are listed here, and others that are not, that were when I was previously in previous years. And engaging those individuals, those sponsors, with your passion and the reason for your passion, and being responsive to that program officer, that program officer is your advocate, as well as you are part of their team, right? They depend on you and you depend on them. And again, for me, that investment from sponsors, whether they're federal sponsors or donors and everything, in foundations, everything in between, is a covenant. It's a covenant between me and them, that I am bringing my best science, my best self, the integrity of our science, to a shared quest. And in this case, in my laboratory, that shared quest is to prevent, treat, and cure Alzheimer's disease. In the Center for Innovation in Brain Science, we have expanded that quest to include Parkinson's, multiple sclerosis, and ALS as part of that grouping of age-associated neurodegenerative diseases. So with that, I will thank you and take questions. So, Caitlin, do you want me to continue to share? No, let's just stop sharing.
Yeah, I'll keep an eye on the chat. So we got one question in the chat that asks if you could just speak a little bit about the importance of diversifying in clinical trials.
Yeah, so that's very important. Now, I will tell you that, so for example, we also did I think I mentioned that we did an analysis of sex difference in our allopregnanolone trial and did not see a sex difference at the maximally effective dose. But we did see a genotype difference. So diversity, I know, can mean a kind of ethnic or racial diversity, can also mean genetic diversity. So in our phase 2 clinical trial, we are very specific about recruiting across the cultures, the Black African American cultures, the Hispanic cultures, as well as the white cultures. So we're very conscious that we want to make sure that what we're developing, just as we did in our phase 1, we want to make sure that what we're developing is, where does it work and where doesn't it? And part of that may well be the racial genetic diversity that comes with one group versus another. Thus far, we haven't seen that because we had people of African origin and Hispanic origin and non-Hispanic whites in our phase 1, small numbers. We just didn't see a signal that said there would be those kind of differences, but obviously you need to test and find out, which is what we'll be doing in our phase 2.
Definitely. We also have another question. It says, have you looked into these patients if they were chronically activated? I'm not quite sure. Souvenir, if you want to jump in and ask your question, I'm not quite sure.
As you have shown that these patients were at the early ages, right? And these patients are subjected to a lot of stimulus, and some of these patients are chronically activated in terms of their immune response.
It's a very important issue, this immune, kind of profile. And what we found, and again, very preliminary, and this is work done by Dr. Archie Mishra in my laboratory, and Nicole Delamonte, or I'm mispronouncing her last name, Nicole, is that the immune system seems to be exhausted. Interestingly, it wasn't quite as functional, right? So they didn't. There was a lot of, essentially, to your point, there was evidence for inflammation, to be sure, like inflammatory cytokines. And then in addition, what Nicole Delatorre and Dr. Artie Mishra did was to ask how functional these immune cells were, and they weren't very functional. Again, very preliminary data. What we were able to detect is that allopregnanolone reduced the inflammatory signals, increased immune signals of wound repair and healing, wound repair and recovery, as well as to restore function to those immune cells. So early days, but it's a great question.
Thank you.
And what kind of points do you have if someone wants to be involved in research, whether it's research that the center is doing or just Alzheimer's research in general? How do you advise people, just the community members, go about getting involved in research and clinical trials and things like that are going on?
Yeah, so there are a couple of, so for clinical trials, there are two kind of broad avenues. One is the Alzheimer's Association has a clinical trial match website. And if you're looking for a clinical trial, they have them listed there, the Alzheimer's Association. The other is through the Banner Alzheimer's Institute. They have a registry for individuals who have family members with Alzheimer's disease. Now, part of that registering as a family member of someone who has Alzheimer's disease registers you, but what you also are able to see is potential clinical trials in your immediate area. The National Institute on Aging, and it's on aging, National Institute on Aging also has a listing of clinical trials in Alzheimer's. And there's a website maintained by the federal government called clinicaltrials.gov. And clinical trials is all one word, .gov. And that will list all of the clinical trials in Alzheimer's. Now, it can be overwhelming, right? And so that's where I think having folks here in the local Alzheimer's Association organization, the Southern Arizona Alzheimer's Association chapter, as well as the Banner Alzheimer's Institute can be very helpful. We will have a link to recruiting for our phase 2 clinical trial of the first regenerative therapeutic here on our website for the Center for Innovation in Brain Science as soon as we're ready to recruit, which we will be probably posting next month.
That's really exciting that you guys are so close. That is all the questions we have in the chat. I do want to give anybody else a chance though. Does anybody want to jump in with a question? Please feel free. All right, well then it looks like that's all, Dr. Brinton. Thank you so much for your time today. And for everybody that joined in, we'll be sending out a link to the recording at the end of the week.
Thanks so much.
Thank
Stay
you.
well. Bye.